Thank you very much, and thank you for the honor to be here. I'm sure that everyone is enthusiastic about taste even before listening to Charles' lecture, but definitely now. This is how we choose what we eat. This is how we choose what's good for us, what's bad for us, and it has a lot of implications for each one of us in the food industry and so on. In particular, I want to focus on the bitter taste, because there are lots of questions in this field, but the questions that I would like to address are how abundant is bitter taste? If I just walk in the forest and take a taste, what are the chances of hitting something bitter as opposed to, let's say, sweet? And also because the bitter taste receptors, which are GPCRs, they come in subtypes in several in families. So it is interesting to understand the interplay between the numerous receptors, the numerous ligands, how the selectivity and promiscuity work, and what do they mean? Why are they important? And I want to also to address the toxicity bitterness relation that Charles introduced yesterday. And in the end, there is a surprise about a surprise sweet tasting molecule, which I would like to share with you. This is a newer research, and I hope it will be interesting for you. So, bitter molecules. So I started working on this several years ago, and would typically read in a paper, there are thousands and thousands of bitter molecules. And I didn't really know which ones, so we decided to set up a database of bitter compounds called the BitterDB. And we took information from publications, from the Merck index, because chemists used to taste what they synthesized back in the older days. And so this information is available from the book of flavors. And we have published the BitterDB, which is the publicly available resource where you can search by the structure of the compound to see if there are any similar chemically similar compounds within the database, and so on. We just published an update where we went from about 500 bitter molecules to about 1,000. We now we use about 140 different publications, and we increased the number of species. So we started with human. Now there is information about molecules that activate bitter taste receptors in chicken and mice and in cat. And there are additional new things in there. So one of the things, looking at this data, which we collected, we didn't produce ourselves, we collected this data. If we look at that, though, we see that some bitter ligands activate multiple bitter taste receptors. So I call them here promiscuous, but what I mean is that they are T2R promiscuous, just in the realm of T2R. And this seems to be kind of consistent across different species. So a molecule that would activate several bitter taste receptors in human also activates several bitter taste receptors in other species. So we wanted to know, first of all, if there are particular physicochemical properties of molecules that are promiscuous versus those that are selective. And indeed, we were able to find such physicochemical descriptors, and we found that the big, flat, and polar molecules tend to be selective, so to have just one T2R receptors associated to them or activate one, while small, global, and hydrophobic tend to be more promiscuous. And that probably makes sense because it's based on like general hydrophobic interaction with not particular, very restricting pocket that has to accommodate it. And we can even predict which additional bitter molecules should be selective, and we could confirm that we are doing not bad with that using an external validation set. Okay. So this is on the side of the ligands. So we have some ligands that activate multiple receptors. We have some that activate only a few. What happens on the receptor side? Here, again, similar situation. Some receptors have many known agonists. Most of the known ligands are agonists. Some are orphan, and some just are activated by very few or single ligand. So if we look at the most promiscuous receptor, the T2R14, one of the bitter taste receptor subtypes, and we look at the ligands that activate that particular single receptor, it looks really crazy because they're ligands that are completely dissimilar from each other, and they still activate the same receptor. And we wanted to understand how is it possible. And if we just quantify the chemical similarity, in blue you see very, very low chemical similarity calculated by Tanimoto coefficient here. In the red, it's identical, and here it's just one little group of homolones, hop homolones that are found in beer and bitter, and these are the only ones that are similar to each other. All the rest are very dissimilar. So one of the questions that we asked, and actually others have asked before, is the binding site, is that the typical family A GPCR orthosteric binding site, is this the same binding site that's responsible also for all this zoo of molecules? Or maybe there are multiple binding sites for agonists. Maybe there is, or maybe it is this canonical binding site, or maybe it's a single site but a different one. And so our approach to answer this question was the following. We tried to use homology models in combination, and this is really crucially important, in combination with experimental studies. All of this work here was done with Wolfgang Meyerhoff and Mike Burns to see whether we can identify the binding sites for the different ligands. So we need to construct a model. We predict potential pockets that could be, in addition to the orthosteric binding site, probably relevant, dock known ligands, and then we predict residues involved in binding, and then these need to be confirmed by site-directed mutagenesis, or also another indirect way of giving some support for the binding site that we suggest is that if we are able to use the model of this binding site to correctly identify additional ligands, then it is not a direct evidence, but it gives some support that probably what we are identifying there could be relevant. So we did that, and I'm showing here just one example. So this is two different ligands, aristolochic acid and picrotoxinin. For this one, we predicted this tryptophan and this one to be important for binding, and the same ones also for the other molecule, which is chemically different. But here, this glutamine was predicted to be of importance. Instead, here it does not appear to play a role, but there is another phenylalanine that is important. And if we are right, yeah, and this is just to show in overlap, so basically it's the same binding site here with part of it overlapping, and then another part is specific for each of the ligands. And if we are correct, then, for example, heterologous cells expressing the wild type or the mutant would be activated by the aristolochic acid, but the activation would be diminished or decreased if we mutate these residues that we found or predicted to be important for binding. And that was the case, and these are the two common ones. But then, if we go for the selective ones or the special ones for each of the ligands, this phenylalanine is important for aristolochic acid, as we predicted, and indeed mutating it is problematic for this ligand. But another one that I didn't mention, and this Q266 is not important, and indeed doesn't do anything to the activation. And here we're comparing to the activation of the wild type by picrotoxinin. The phenylalanine doesn't care. Here it's the glutamine, it diminishes. So this was one direction. As I mentioned, site-directed mutagenesis supports the predicted binding site. And the other one is if we look at the binding and we create a pharmacophore model which tells us what should the ligand bound in this binding site look like. And here we represented the binding site routed as excluded volumes, right? So we represent the presence of the pocket by excluded volumes. And then we can screen for libraries of compounds, which we did. And we look for those that fit into this model of how the ligand should fit into the receptor. And indeed, three of these four predicted, of the four that were experimentally tested, you see here in green, indeed, activation of the same receptor of T2R14. This one was a mistake. It is cocaine. It's cocaine. It is bitter. And Mike was really unhappy because he had to have it under like thousands of logs and a lot of regulation because it's cocaine, and he had to test it for that. And anyway, but it does not work through this receptor. Similarly for T2R10, I won't go into a lot of details, but just to show that the same residue could make an interaction that is hydrogen bonding, and then if you mutate to threonine, it's fine, but then if you mutate to valine or alanine, it's not good. But for another ligand in the same binding pocket, which is not using hydrogen bonding to that specific residue, actually you prove by mutating to alanine, threonine is not good, and also valine is not good. So our conclusion was that indeed it seems that this binding site is co-optimized for binding different ligands in different ways. So basically, of course, it does not exclude the existence of additional potential binding pockets, but at least from what we saw in several works, the main way of binding is through the canonical orthosteric site of, like, similar to family A, but there are different subsites within this binding site, and there are different types of interactions with different ligands, which allows for accommodating multiple dissimilar agonists. So then we said, okay, but if this is the same major pocket, could we maybe learn something about the pockets of the binding site pockets of GPCRs and be able to predict, just looking at the binding pocket, how many ligands it would be able to accommodate, or actually not how many, but rather how diverse they could be. Can I predict the promiscuity just from the physical-chemical structure properties of the binding pocket? And so here on the left are libraries of known ligands for the GPCRs for which structures were known then. And we calculated different properties. So for example, volume, exposure donor area, and acceptor area, free energy of water sites, because actually when we started doing this, I thought that there are placeholders in the binding site who are sitting in different places in the binding site, and then can be kicked out as needed when different ligands come in. How many disulfide bridges are there, the properties of the amino acids, accessible rotamers to give an essence of flexibility, the length of secondary, the ECL2 loop, and so on. And then we did a statistical analysis, which basically from all these properties found the ones that contribute to the known diversity of the ligands. So the winners of this contest were the surface area of the binding pocket and has to be high, and the hydrophobicity has to be high, but the donor area has it's a hydrogen bond donor area or number has to be low in order for this pocket to be promiscuous, right? And again, it makes sense because if it's hydrophobic and there is less directionality, if there are less hydrogen bond donors, you can accommodate many different things. So exposure, hydrophobic content and donor area down, these two up, and you get a more promiscuous pocket. And similarly, it was done for the different T2R subtypes, because there we have a range between them, the more and less selective, and we did that. And in fact, we were even able to predict the expected number of observed of ligands just based on the structures that just came out after we did this analysis, so these additional crystal structures that were published. I know we have to repeat it now because there are much more. And there was an excellent correlation between what we predict just based on the pocket nature to what is known in terms of ligands. And it could even be applied to homology models, but it would not result in a very beautiful correlation like here, but still can be used. Okay, so now we know what are the physicochemical properties of selective and promiscuous receptors and selective and promiscuous ligands for T2Rs, but now we wanted to know whether the selective ligands or ligands that only activate a single T2R, is that single T2R also a selective one? In other words, is it reciprocal on the side of ligand and the side of receptor? And the idea would be that if specialists evolve after generalists, right? So we would have like promiscuous T2Rs, and from these would evolve those that are special for particular ligands, and these ligands might be those that drove the evolution of this binding site that is special for the unique ligand. That was the hypothesis we wanted to check, and if that would have been true, we would, if we order the receptors, the T2R receptors like this, promiscuous on the left side, selective on the right side, and then the compounds are also ordered from promiscuous, so activating many T2Rs, to selective, activating only a few T2Rs. And if this hypothesis holds, we should have something like this. So the selective compounds mainly activate selective beta receptors. But actually what we found was different. We found that most of the selective ligands, so the ones that for which we only know one, or only one T2R is activated, most of these T2Rs are promiscuous. So it could have not been something that drove the evolvement of this receptor. So most selective compounds activate T2Rs that are promiscuous, and this is what I showed you before in cartoon. This is really numbers, and this is numbered, colored by how many receptors are activated. And you can see that very few green ones appear here. Everything is happening for the promiscuous receptors. So, okay, so there could be two things. First of all, maybe this hypothesis is not so relevant. For example, perhaps it's the combination of different T2Rs that is of essence, and that's what's important. It's not just having a single T2R recognizing a ligand. Maybe it is important which ones work together, maybe. Or maybe we just didn't find the relevant unique ligands, right? Because we are working with what we know. Maybe there is a very important ligand that was the reason for that receptor to evolve the way it is. We just don't know that ligand yet. So to kind of start tackling this, I'll show a couple of things that we did, and then I'll show you how we are working on discovering additional ligands. So one thing we wanted to check is whether, if you have a ligand that activates multiple receptors as opposed to a single one, is it going to be more potent or more aversive or more disgusting in vivo? And the easiest way we found to do it was to work with chicken, because chicken have only 3 T2Rs. And also I'm at the Faculty of Agriculture. And so we looked at these unique ligands activating just one chicken taste receptor. This one, we had 2 ligands for it. And then we had quinine, which activates all 3. So you can see here, but I will show you also visually, that there is actually no simple rule. So basically, if we look at the ratio between in vivo threshold for being averted from the solution that contains the compound, the in vivo to in vitro ratio is just different for different ligands, and the one that activates 3 different subtypes was actually not more effective was not more disgusting to the chicken than others. So of course, this is the beginning. Probably much more needs to be done, but it does show that there is not a clear answer for why there are multiple receptors. Another thing we wanted to check was whether maybe multiple receptors serve as backup for toxic compounds, right? Because if bitterness signals toxicity, if something is really, really toxic, it's really, really dangerous, it would be beneficial to have several backup receptors to recognize this this compound. And so something happens to one receptor. So it was already shown before, many years before. John Glendening showed that there was no correlation, but now we have much more data, so we wanted to check it again. And what we wanted to check is whether if we plot the number of activated T2Rs versus the LD50, so the more toxic here, the less toxic to the right, higher LD50s, we did not find a correlation, and this is the same picture, just here non-toxic compounds and some outliers taken out, but in any case it doesn't look like the more toxic you are, the more subtypes recognize the bitterness of that ligand. Okay. So right. So we don't really know yet what is going on with these multiple subtypes, but we do want to be able to predict and identify additional ones. And we can do it in, by several methods. One is ligand similarity to a particular bitter ligand, right? So if I have a particular bitter ligand, or like I have a key to a door, I can look for similar keys. A ligand-based approach without knowing anything about the binding site on the receptor. The other approach is to fit ligands into the binding pocket that we have identified before, and see what else can fit in there. And the third one is a machine learning approach, so basically taking a compound, looking at the structure, and then saying if it's going to be bitter or not. So it is interesting, just I want to tell you, it not only to answer this question that we had before about this unique ligands for unique compounds, if they exist at all, but also because taste receptors have been shown to express extraorally. And there's a lot of work by other groups showing A, their expression, and B, their relevance for different indications. And I will just mention one thing, even though there are many more. So Noam Cohen showed that T2R38 polymorphisms underlie susceptibility to upper respiratory infection. So basically, if you remember from a childhood lecture yesterday, you can be a taster, non-taster, or super taster. And it turned out that if you are a non-taster, you may have a higher chance of having chronic rhinosinusitis, like infection in the upper respiratory infection. And they have shown that this is due to the fact that if you are a taster, then the receptors, the tasting, the functional receptors that are expressed in the upper airways respond to molecules secreted by bacteria. And then downstream, they actually release NO, nitric oxide, and kill the bacterial part of it. And this is how it fights the infection. If you are a non-taster, the bacteria secrete these molecules, but you're not feeling this and nothing is happening. So one of the things one would want to do is first of all, identify additional molecules that could activate this, right? Because then you don't need for the bacteria to produce a lot of squirm sensing molecules and to activate the system. And the other thing, it would be nice eventually if it would be possible to find something that activates the non-taster version. If that exists, then you could also treat people who have the non-taster polymorphisms. But let's just start with the taster, okay? We know some ligands, and interestingly, these ligands, typically the most active ones, contain the isothionate moiety. And we have true negatives and true positives, and we can basically, and by the way, the glycosinolates in broccoli, for example, or in other vegetables, release such molecules, and this is why some people really hate them and some people don't mind. Anyway, so we went to Antonella in my lab went ahead and screened all the BitterDB and the drug bank, the experimental as well as approved drugs, to look for these substructures and then for some molecular descriptors of active molecules and the pharmacophore that would be suitable for these type of molecules, and ended up with several compounds. Only 4 were purchased and tested, and they were tested on primary human sinusoidal cells in Noam Cohen's laboratory. And here, indeed, we expect that if this is a primary cell from a human who has the taster version, these molecules would activate. This is, again, a no release. And if it's non-taster, he would not react. And this is exactly what happened for all of these molecules. The other approach, I will only briefly mention this. So the other approach is structure-based discovery, but I just want to say that it's very difficult because what we are dealing with here are receptors that have very low similarity to other GPCRs. So basically like 30% in the TM regions, similarity, not identity at best. And they have a lot of the hallmark motifs of GPCRs that are either not conserved at all or changed a little bit and have something else, something different from other GPCRs. So it is not even trivial to be able to use homology models based on something so remotely similar to perform for identifying new molecules. However, as I said before, it is really crucial if you have a very integrated prediction and experimental conformation, and then if needed refinement, it can work. So here I'm showing a new work that we started from a homology model that was already performing pretty well, that I showed you a little bit before. So we knew that it seems to have the binding site at least in the right position. But here we started from one ligand, one agonist that was pretty good, one of the best that we had for this receptor, flufenamic acid. And in collaboration with Gima in our lab, basically did the following. Screened, so there are particular building blocks that are easy for them to make the chemistry with. And so multiple molecules were created in silico, and then Antonella docked them, these 1,000 virtual molecules, into the model that we had already. And then they were tested in a Barron's lab. And basically, so here I'm showing some of the results. So you can see there are some modifications that do well and some render the molecule inactive. So overall, it's great because just from 20 tested compounds, we found 12 agonists and 5 agonists even more potent than the flufenamic acid that we started from. But nevertheless, there were 4 inactives. And that was a little annoying because we screened, so only those molecules that fit really well into the binding pocket that we had at that time were synthesized. And nevertheless, some of them were inactive. So we thought, okay, now we have the opportunity to optimize even further our binding site, and that's what we did. You can see here, well, maybe it's too small to see, but you can see that the original model we started with did not discriminate between what eventually turned out to be true actives and true inactives. It basically scored them all the same. But when Antonella refined the molecule, refined the model, we have an excellent ROC curve. So we have a model that really gives good score to real agonist and bad score to the inactive molecules. And so now this refined model can be further used to screen additional data sets. And this is here. So, okay, so this is encouraging. So despite low homology, low similarity to templates, we still can work if we pay a lot of attention to experimental data and incorporate it. But this is really painful, right? If I want to do it for each individual receptor, there is a lot of work, a lot of manpower to understand how the binding site looks, and even then we will find some new ligands maybe, but we cannot say about something that does not fit into our refined and excellent model. If it doesn't fit in there, I don't know what it means. Does it mean that it just binds in a different way, or does it mean that it is not bitter? And we do want to know what is bitter or not. We kind of do want to have a classifier between bitter and non-bitter molecules. So then we decided to go for machine learning approach, because we have already, well, when we started, we have about 700 molecules that are known to be bitter. If we have enough molecules that are known to be non-bitter, we can apply these techniques to just sort them into bitter and non-bitter, without committing to which receptor is it, which ligand is it similar to, and so on. Now, just to say, it's not so easy, because in green, you see on this PCA, the principal component analysis of the chemical space, the bitter molecules have different. This is mainly dominated by molecular weight, and this is mainly dominated by hydrophobicity. So you see green everywhere. You see random everywhere. But we don't know if random means non-bitter or not, right? We actually, my first question, what is the percentage of a random library that has a bitter taste? We still don't know the answer to that. So, the most important thing for machine learning approaches is to have good input, so true positives and true negatives. And the true negatives we started from were non-bitter flavors. So this is actually very confined. They don't describe all this chemical space. Anyway, I now worked really hard and developed the non-bitter set. And I will skip through this quickly. This is the method she used is AdaBoost. So it's basically like a forest of decision trees which are trained on misclassified data. So together they eventually give good prediction, good classification. And these are the data on training set, excellent, but on test set it's also very good. And then on external validation, which is important because we don't want to overfit, it is also doing good. And then we did also additional testing, really prospective prediction. And anyway, we can say that it's 17 or 90% accuracy in the bitter prediction. Okay, so now we can maybe get to the question of the relation between bitterness and toxicity. Because I always also say bitter is to protect us from poison. But we wanted to kind of, quantify, is it really, what is the relation? And it is true that some poisons are bitter, and some bitter are toxic, but there are also foods that taste bitter, and actually are healthy ones. These are the examples of compounds that give the bitter taste to these foods. So, okay, so what is going on, and how can we quantify it? So what Ido did was the following. So we have the bitter DB. At that time we had close to 700, and he took a data set of toxic molecules, and he wanted to know whether, now that we also have the bitter predict, how many of these are going to be toxic. And we did it for several, data sets of compounds, the food DB, the compounds that are found in food, the drug bank, natural products, and so on. So our expectation when we started doing this was that in the FOCTOX is the library of acutely toxic oral toxicity. So that would be perfect, right? This is exactly what we want to be able to detect by bitterness, because these compounds that are taken orally are acutely toxic, So we would expect them to be very bitter, and maybe combitox as well, and then the other less so. But that's not exactly what happened. So actually in the natural products, we had a very high percentage of bitter predicted compounds. In FoodDB, we had quite a few. And here it was not huge. And if we do an even more stringent, if you want to be even more sure in the prediction of toxicity, still, okay, these are not the most bitter enriched libraries. Okay. So first of all, we know there's still a lot of probably bitter molecules, including Chebby. It was representing like random compounds, so including there. But and the natural products are and approved drugs are enriched in bitter, but many toxic compounds are not predicted to be bitter. So we are also checking now for developing a predictor of intense bitterness, because here it was just yes, no answer. Is it bitter or not? Okay. So what we found is that there is an overlap, of course, between toxic and bitter compounds, but it's only partial. So we found that only about 60% of combitox compounds are predicted to be bitter. And also we find that 60% of bitter compounds have documented toxicity. So here again, these are non-bitter compounds, and these are the distributions of toxicity of LD50s. And it looks like this. In the non-bitter compounds, only few are really fatal. Then we have toxic, harmful, and more maybe harmful or non-toxic. So now showing the same histogram for the bitter, I would expect exactly the opposite trend, right? I would expect that most of them are at least toxic, because if it's really fatal, maybe there is not enough, there is not a chance in evolution to develop the ability to sense it, but they should be toxic and harmful and less. Okay, but that's not what we found. Again, the trend, it's not exactly the same, but the trend is like that. So there are not a lot of LD50s known, so maybe the picture would change with more information, but according to this information, this is what we have. Okay. So, but I want to mention, though, that if we think about the ability to taste bitterness and by herbivores, and the need of the plants to produce the compounds, in fact, it may be enough for the plants just to produce something that is bitter. It doesn't actually have to be toxic, right? Because if it saves you and you're not eaten, that's good enough. So it might be that, so that might explain maybe that why the bitter taste is aversive, even though it is not always toxic. Anyway, because of the lecture yesterday from Charles, I wanted to show this. I didn't plan it, but I decided to add it, because we wanted to test this hypothesis that if bitter taste is supposed to warn against poison consumption, it should elicit some negative emotions, suspiciousness, and distrust. Like very, naively, I was saying, like, if somebody is trying to poison me, right? I'm sensing something bitter. How should it affect? And we did the experiment, which was zip and spit. It was of bitter solution, followed by seemingly unrelated assignments. Some of the assignments were financial risk-taking. I'm not showing this because nothing came out significant. But the PANA score, which is the measure of immediate emotion, did come out significant. And this basically, it's a simple score where you just rate the emotions, which are the positives like interested, excited, strong, enthusiastic, and so on. And then you have the negative, guilty, upset, distressed, and so on. And we wanted to know if taking a zip without swallowing of bitter compound or bitter solution would affect it. And indeed, we found that overexposure to quinine results in lower PANA scores significantly. So it's the same person with that person's score. And if we repeat this with props, so it's like PTC, where you have tasters and non-tasters, we see that the non-tasters like the solution more and also don't have a strong effect on their mood, However, the strong tasters dislike the solution and also have a decrease, less significantly, but still significant in their immediate mood. And this is why I wanted to show you, though, because then we did it for not as affecting the same person, but just we took different groups of people. One group tasted quinine, another group tasted sucrose, and another one tasted a mix. And we actually thought that with the sucrose it would improve the mood, but it did not. And I remind you that it was just dip and spit. There was no swallowing. And so I think, I mean, it's interesting that the effect is asymmetric, because with the sucrose solution we don't improve the mood. But, well, this is often in life, of course. It's easier to disrupt something than to improve. But with the quinine, the PANA score goes down. All right, anyway, and so now the surprise molecule that I promised you towards the end of the talk. So we got interested in sweet taste. And this is just a depiction of some of the things people have discovered about sweet tasting molecules, which include agonists and proteins and enhancers. This is from Phenomics. So it's a heterodimer, right? And here the orthosteric binding site is actually in the VFT domain, like in the glutamate receptor. But in the monomer that is common to both the sweet and the umami, there is also a binding site which is similar to the family A orthosteric site, but here it's allosteric, and it can be used, for example, cyclamide as a sweetener that works through this domain, and lactazole is an inhibitor that also binds there and inhibits the sweet taste. All right. So, what is the surprise molecule? The Nobel Prize laureate in chemistry in 1934. Uri, he got it for the discovery of deuterium in 1934. In 1935, he responded in a letter to Science to some reports that people have mentioned the taste of deuterium oxide or heavy water. So he and colleague decided to check what's going on there, and they did an experiment where they tasted the regular and heavy water. It was just the two of them, so it's N equals 2. And they said that there was no difference whatsoever in the taste of the two types of water. Okay, so some people have been looking into it, and because D2O is used a lot in NMR, there are many NMR people that get to taste it, and there are YouTube videos showing that actually it has sweet taste. Anyway, a colleague of mine, Pavel Jungwirth, said that they have been playing with it for some time, and they want to understand the sweetness of that water. Now, because of a different story, I don't have time to go into it, but it's really, really important if you do something like that, which seems kind of unbelievable, right? Why just a tiny change in water, so it's just a tiny change in the strength of hydrogen bond due to tiny change in the mass of deuterium relative to hydrogen. Why should it change the reaction of, we don't know yet what, receptor or channel or anything, but what? So you should be very, very careful to make sure that it's not an artifact and you're not comparing something that got into the D2O and it's not there in the H2O, and this is why you're feeling something, but it's not the effect. So we've been working on ultra pure waters, both D2O and H2O, so very, very high purity, purchased and then distilled 4 times. And here you see that the D2O distilled 1, 2, 3, 4 times. This is the sweetness measured on particular on a scale by people. So here are 28 subjects tasting that, and you see that all the purifications of H2O are always less sweet than the D2O. is it smell or is it taste, right? So there's Natalie did another experiment, which is a triangle test. You basically have 2 H2Os, 1 D2O, and the panelist needs to choose the correct one, and you know which is the correct one. And you can do it just with the smell without tasting. You could do it just with the taste by closing, by nose clips, and you can just do it just tasting without closing the nose clips. And that was done for 25 people. So only 9 out of 25 chose the correct sample, and this is just random because 30% correct answers is just random. For taste only, it was significantly better. Smell and taste was even better, but we can conclude from here that taste is enough to discriminate between H2O and D2O. We can also show that if you mix D2O with H2O, the sweetness is, here again 30 panelists, the sweetness is in dose response, so the more D2O you have, the sweeter it will taste. And it is additive to sucrose, so not exactly additive, because at the higher concentration we tested, it seems to kind of converge. But at least at mild sweetness, it adds, so the D2O, here we have increasing sucrose solution, one time in H2O, the other one in D2O, and you see that D2O is ranked as sweeter on average by, again, 30 people. And this is the same experiment shown again, but also now we did with glucose. And again, the D2O solution of glucose is sweeter than in H2O. And it does not happen in the bitter solution, right? So just as a control, if we take quinine, so here you see the black ones are the D2O. Here the black one is D2O, and it is less bitter. Here we're testing for bitterness. It is less bitter than the quinine alone. And this is just to confirm that we're not looking at some effect that just makes everything stronger, but it specifically makes gives a sweet signal. Okay. And so now remember I told you about lactazole that inhibits the T1R2, T1R3 receptor. And what we could show is that adding lactazole to the D2O actually diminishes its sweetness, and it does nothing to the H2O. So this is our first hint that it is the T1R2, T2R3 that is involved in sensing the sweetness of D2O. So it's a likely candidate. How it works, we still need to investigate. But if we schematically present this here, right, if there are some conformations of the upper receptor, we expect stabilization by D2O in the active conformation. And that can be done by several not mutually exclusive ways. One could be that it is stabilizing the VFT binding domain in a more active conformation. The other one could maybe do something with the orientation of the different monomeric domains, one with respect to each other, and maybe in the presence of D2O, this is stabilized. And this is, it has been shown before that that D2O induces folding and compactness. So it could be this case, or it could be particular water in the TM domain that are, when it is regular water, it's just there, and there are crystallographic waters that were found, and not only found, some of them were even conserved across different structures. But maybe there is, when D2O comes in, there is a network of stronger bonds that that is created there that leads to activation, somehow maybe related to the way that cyclamide does it in the TM 3 in the transmembrane domain of T1R3. So just to summarize, we wanted to know how many bitter ligands are there. So we know 1,000 now from literature, and they can be searched through BitterDB. And we have predicted, well, actually I think it's even more than tens of thousands. I think it's hundreds of thousands that can be tested. We were really bothered by how they can be recognized by the receptors because they are so diverse. But we found that actually orthosteric site can do a lot of tricks, even though, of course, we cannot rule out that there are additional binding sites, but the orthosteric binding site can be adapted to or is co-optimized for multiple ligands. Then we were concerned with the selectivity promiscuity question, and so we could discern the physicochemical features that make a ligand or a receptor more prone to promiscuity, let's say, but we still don't know what are the physiological roles of having, or is there a difference of having a ligand with multiple target T2Rs versus one with a single one. We can predict ligands computationally, and we can do it by ligand-based, structure-based, or machine learning approaches. And this is not, especially the structure-based is not trivial because we are starting from really low-resolution models, but it can still work. The bitterness toxicity quantification, we found that there is a partial overlap, but it's probably not, so not everything that's bitter is toxic, and not everything that's toxic is bitter. And I had this little detour to show the taste effects on mood, which was asymmetric. We found that bitter affects mood, but sweet actually, without swallowing in this case, but sucrose did not improve if you don't swallow it. And then finally, I showed our results suggesting that this is a sweet taste, sorry. It elicits sweet taste. That I think we have shown, I mean, with this we're sure, and pretty positive because of the lactazole experiment that the target is T1R2, T1R3. And sorry for the typo. And I would like to thank my students, many of whom are here, and Mickey and organizers, and I'm happy to take questions as allowed.